Good morning. This is Julian speaking. I can see that there's already a lot of people have already logged in. We'll wait another minute to make sure everybody is ready, since there's still people joining the webinar, and then I will start the presentation. Okay, welcome everybody to today's webinar. My name is Julian. I'm the CSO of Cytina, and I'm really excited to host the third webinar of our Selling Cytina webinar series that we have in April. In the two previous webinars, we talked about setting up efficient cell line development workflows and about the isolation and cultivation of microbiome cells from gut samples. Now I'm super excited to present you another workflow for efficient generation of single-cell RNA sequencing libraries. So thank you very much for joining. The presentation will last around 30 minutes, and then we will have some time to address questions. For those of you who use the GoTo plugin the first time, there's a couple of options that you can access by clicking on this arrow here. Please note that everybody in the audience is muted. You are, however, able to submit questions using the text box down here. So please submit your questions whenever they come up. We will collect them and address them after I went through the slides. We also uploaded some additional material, a brochure on the X-Site instruments and also on the IDOT. That you can download throughout the webinar. Let me briefly introduce you who we are and what we are doing as a company. Since last year, Cytina is part of the Selling Corporation, a young company that has grown rapidly over the last years. Founded in 2016, so we are now present in over 60 countries with Selling, and we have offices all around the globe to develop new products and also to support our customers. Our product portfolio covers a number of different areas within the life sciences, and we provide tools from bioprinting to liquid handling and single cell handling and isolation, as well as imaging. In this webinar, we'll discuss a workflow that is enabled by the IDOT, a low volume dispenser, and the F-Site for isolating single cells. Here at Cytina, we develop instruments for automated isolation of single living cells since 2014. All our instruments are capable of label-free cell isolation and come with image-based cell detection. It started all off with the SCP that you can see here that was launched in 2015, and in 2018 we launched the X-Site series comprising 3 instruments: the F-Site and the C-Site for isolating mammalian cells, and the B-Site, which comes with a higher resolution optical detection system for isolating microbial cells such as bacteria or yeast. Together with the company Dispendix, which is also part of the Selling family, we can now offer also a highly flexible low volume dispenser, the IDOT. In fact, if you look at the largest pharma companies, most of them are already using our technology for single cell isolation to either generate cell lines or analyze single cells. This slide provides an overview of the application areas that are addressed by our customers. Let me briefly go through this before we start with today's topic. Our technology is very established in cell line development, which typically refers to generating mammalian cell lines for the production of therapeutic proteins, which is a very regulated workflow. Another very exciting application is the cultivation of single bacteria cells obtained from mixed microbiome sample, for example, to study gut microbiota. These two topics has been addressed in previous webinars. The third application is single cell analysis and most prominently single cell genome and transcriptome sequencing, which is the topic of today's webinar. Let me briefly go through the agenda. First, I will provide a brief introduction on single cell RNA sequencing. For those of you that are not yet familiar with this technique, I will then introduce you to our technologies for single cell isolation and for low volume liquid handling. Finally, I will present a workflow for efficient preparation of single cell RNA-seq libraries at significantly reduced reaction volumes. Single cell RNA sequencing is currently the most widely used single cell omics assay. And one reason for this is this increased popularity is that there are many technological improvements that came up over the last 5 years. Improvement in chemistries, in microfluidics, and also in data analysis. But also the fact that single cell transcriptome analysis now allows to look at heterogeneous sample in much more detail at a routine level. And as depicted here, it often starts with a complex sample, for example, a tissue sample. First, the cells need to be brought into suspension before they can be isolated for subsequent amplification and sequencing of transcripts present in individual cells. After sequencing, the reads obtained need to be mapped to a reference genome to compute a count table as depicted here. In addition, typically several filtering and normalization steps are applied to improve the data quality. Based on the count table, we can then look at differential gene expression or cluster cells based on their expression patterns. Common applications of single-cell RNA sequencing include the identification or characterization of cell type population. Using full-length transcript sequencing lets us also look at differential splicing events in single cells or allele-specific expression. And we can also investigate processes that are time or cell cycle dependent. The power of single-cell RNA-seq, along with increased robustness of the methods, already contributed tremendously to a better understanding in a variety of different disciplines, including developmental biology, oncology, and immunology. But we also see that these methods are being picked up now by the pharmaceutical industry as an additional tool for developing new therapies and drugs. One very important aspect for precise single-cell analysis is a robust workflow for generating the sequencing libraries. And it's widely accepted that automation increases robustness and reproducibility since it reduces operator bias. Finally, it's important to set up cost-effective workflows since the number of cells to be analyzed is typically much higher compared to any bulk sequencing experiments, and the reagents needed to generate NGS libraries from minute amounts of target molecules present in single cells are often expensive. Considering these prerequisites, we have decided to offer an affordable and open platform based on proven technologies for single-cell isolation and liquid handling. Before I discuss some of the data that you can get from this workflow, I want to provide a few more details on the two instruments. Here you see the F-side, our instrument for label-free cell isolation and fluorescent-based sorting of mammalian cells. It's a benchtop instrument that's very easy to operate compared to other technologies such as flow cytometry. If you zoom into the printhead here, you can see the nozzle of one of our disposable dispensing cartridges, and we use an optical detection system that's similar to a bright field microscope that detects the cells in the nozzle of our dispensing cartridges. In addition, the F-side comes with a laser and an additional camera that allows us to sort cells based on fluorescent markers. And only those droplets that contain a cell with the right properties are then deposited onto the target substrate. Empty droplets or droplets that contain multiple cells are removed with a vacuum suction that is positioned directly below the dispenser. This allows us to rapidly dispense droplets with single cells into well plates, as you can see now in a short video that I will start in a second. So the video shows the C side, but the handling of the F side is very similar. You open the dispenser, you pipette your cell sample into a new cartridge that is then attached to the dispenser with two screws. We have a pipette tip that we can immerse into the reservoir that gently mixes the cells to prevent them from sedimenting. You then insert your well plate. For RNA sequencing experiments, this would be a PCR well plate with conical wells, typically pre-filled with lysis buffer. Then you set up your workflow and start the run, and the dispenser will go from well to well and dispense a single droplet with a single cell. And after a few minutes, you can proceed with the lysis step or cultivation of the cells. So let's go back to the slides. You can again see our one of the disposable cartridges that you have been seeing in the video. All our instruments for single cell dispensing operate with sterilized single-use cartridges. In fact, the cartridge is the only part that gets into contact with the sample. And this means there's no risk for cross-contamination and no cleaning procedures required. And you can process basically any sample volume starting at 5 microliters. Recently, we have equipped the F side with a module for automated dispenser offset compensation. This system measures the droplet impact position to make sure the droplets are deposited into the center of conical PCR wells without the need for any manual alignment procedures. With 200 picoliters, the droplets produced by our systems are super small to not impact the biochemistry of scale-down reactions, and 96 cells are typically dispensed within 3 to 5 minutes. For each cell that is dispensed, several images are stored to the hard drive to document the cell isolation process. You can see one example here, where you see the cell approaching the nozzle. Here it is in the nozzle. There's another image showing an overlay of the image processing, and the final image which shows the empty nozzle after the cell has been dispensed. Once the fluorescent laser on the F side is switched on, additional images from the fluorescent channel are also stored to the hard drive, along with these bright field images. Now I want to show you a brief video to highlight how the software of our systems look like and how simple it is to operate them. You typically start with selecting your plate format. In this case, it's a 96 well plate. You can add a plate ID or name, then select the wells that you want to address with cells. We have a dot plot here that you can see on the right that helps you to select the right gating parameters. On the C side, this is the size and roundness of the cell, and on the F side, you can in addition sort based on fluorescent intensity. You can look at all these parameters into the dot plot to decide which population you want to target. Once you're set, you can start the dispensing. The software lets us confirm that we set up everything correctly, and then the single cell dispensing starts. On the top right corner, you see a live view of the nozzle of the dispensing cartridge. And you can see cells approaching the nozzle where they are detected and then dispensed into the respective wells. And the progress can be monitored by looking at the plate here. Each time the check mark occurs, a single cell was dispensed and the printhead moves to the next well. And also, let us now go back to the slides. Each time a single cell is dispensed, as you just saw in the video, these image series are stored, as I already mentioned. Now let me introduce you to the IDOT, which is a benchtop instrument for low volume reagent dispensing. The IDOT technology allows to generate droplets using a well-controlled pressure pulse that is applied to a liquid well with a small hole on the bottom, as you can see in the schematic on the slide here. It comes with 8 such individually controlled pressure channels, which generate droplets within a range of 8 to 50 nanoliters. Volumes in the microliter range can be easily addressed given that droplets are generated with frequencies of 100 Hertz. Something that really sets the IDOT apart from other low volume liquid handlers is the disposable source well plate, which holds up to 96 different reagents. It basically looks like a normal 96 well plate that can be filled with standard pipettes or multi-channel pipettes or even pipetting robots. The system can dispense from any of these 96 wells into any well of a target plate, which allows to perform even the most complex liquid handling tasks within minutes. Over the last years, a variety of different protocols for single-cell RNA sequencing have been suggested. Optimization with respect to sensitivity, robustness, and cost-effectiveness is still ongoing, and I believe that we will see further improvements of these assays in the next 2 years. A very popular and well-established protocol is SmartSeq2, which was published in 2013 by Simone Picelli and others. In contrast to droplet-based methods, SmartSeq allows for full-length transcript sequencing. Here on the right, you see an overview of the individual steps of the protocol. It's a figure of the original publication. After cell lysis, the mRNA is converted to cDNA. By reverse transcription, the cDNA is then amplified by a PCR. After purification step, a method called segmentation is used to fragment the cDNA and introduce sequencing adapters. Subsequently, each single-cell library needs to be barcoded by a final PCR before it can be pooled and sequenced. It has been shown that SmartSeq is one of the most sensitive protocols. That's why it's also one of the methods selected by the Human Cell Atlas, one of the largest initiatives utilizing single-cell RNA sequencing to map human organs and tissues. Several labs have shown that SmartSeq2 libraries can be generated in 96 or 284 well plates in an efficient manner, also at reduced reaction volumes. Often, however, labs do not have access to appropriate liquid handling equipment and struggle with complex instrumentation for cell isolation. Here you see the entire workflow that we offer to address these challenges, starting from the single cell sample to the sequencing data. And before I continue to talk about the details of how we set up and validated the workflow, I want to quickly start a poll question. So now there should be a question popping up. Which method are you using or are you planning to use? Are you using or planning to use single-cell RNA sequencing in general? Are you looking into other multi-omics methods, such as, for example, methylation or gene expression at the same time? Or are you looking into single-cell genome sequencing to study mutation in genes or copy number variations? Or are you looking for something else? Please take a second and address this question. I will then show in a few seconds. I will share the results. Okay, let's wait 10 more seconds before I share the results. I can already see that more than 50% of the audience is either using or planning to use single-cell RNA sequencing. Okay, this is the results from the poll. 57% are looking into single-cell RNA sequencing, 38% into multi-omics methods, which is quite a lot, and I think this will be increasing in the future, and almost 20% are interested or are performing single-cell genome sequencing. All right, then let's continue with the presentation. This is again the overview of the workflow, and I will now go through some details before I will show you data. For this validation study, we used a hex cell line that was diluted to 0.8 million cells per milliliter prior cell isolation with the F site. We used an F site with the module for automated dispenser offset compensation, and the cells were dispensed into 384 well plates, pre-filled with 1 microliter of lysis buffer. All liquid handling steps were performed with the IDOT liquid handler. The first part of the protocol, we used a commercial version of the SmartSeq assay from Takara, the SmartSeq version 4 kit, to generate the cDNA libraries. And the second part, the NGS library preparation, we used the Illumina Nextera XT kit. All reactions were performed at 10-fold reduced reaction volumes compared to the original protocols. Here you see a list of the reagents and the respective volumes, both for the original protocol and for the miniaturized protocol. Cell lysis was performed in 1.05 microliter volumes, and as you can see here, the F site hardly introduces any additional volume when the cell is dispensed, since the droplets of the F site are only 200 picoliters in size. The final PCR to amplify the cDNA was performed in 3 microliters. Similar, the Nextera XT reactions were scaled down, and the tegmentation was performed in 2.5 microliters, and the final PCR to amplify and barcode the libraries in 5 microliter reactions. All reagents were dispensed automatically using the IDOT and 1 IDOT pure plate. This results in a 10-fold cost reduction of the 2 most expensive parts of the whole protocol. This slide highlights the kind of data that you get for each individual step. As I mentioned earlier, we obtain images of each individual cell that is isolated with the F site. Here are some examples of hex cells dispensed. The F site also allows to dispense empty droplets from the cell sample for negative controls, as depicted here in the 4th image. After reverse transcription, the cDNA is amplified, resulting in cDNA libraries from single cells, as depicted here in the center, and tegmentation and another PCR to bring in the sequencing adapters then results in the final single-cell sequencing library that can be seen here on the right. These libraries can be then pooled into a single tube for sequencing. Here again, you can see such cDNA libraries for 9 single cells and a pool of 10 cells and a negative control. These are results as obtained from Agilent bioanalyzer that we used to determine the fragment length distribution of the cDNA libraries as a quality control. In general, we observed very consistent cDNA fragment size distribution across wells, and as you can see, the control without a cell resulted in no cDNA. One thing that you can do with the images that we obtain from the F-site is to link them to the data generated throughout the assay or to the final sequencing data. Here's one such an example. Here we looked at the cell size as determined by the F-site versus the cDNA yield after the amplification reaction. And it's quite nice to see that there's indeed a good correlation between cell size and transcript abundance in the single cells. For the analysis of more heterogeneous samples, this might be a helpful tool to improve normalization or clustering of the data. Finally, we pooled 125 cells and sequenced them on an Illumina NextSeq instrument. As you can see here, we obtained high-quality sequencing data. More than 94% of the reads mapped to the human reference genome, and on average, almost 10,000 genes were detected in each individual cell. Here the cells were downsampled to 1 million reads per cell. This demonstrates nicely that our system can be used to generate high-quality sequencing data from single cells. Since the entire protocol is based on 384 well plates, this is really scalable, and such a setup should allow for the preparation of at least 1,000 to 2,000 single-cell libraries on a single day, if you streamline the workflow. Now I want to take the time to highlight 2 slightly more complex liquid handling tasks and how we efficiently address those with the iDot. The first one is indexing of NGS libraries, and the second one is the normalization of DNA samples. Both are part of the SmartSeq protocol that we used here. The ability to index NGS libraries with unique DNA barcodes really provides the basis for high throughput sequencing and is especially important for single-cell sequencing. To efficiently barcode libraries, dual index schemes are very common, as the one depicted here. To barcode 96 samples, 12 forward and 8 reverse primers need to be combined such that each library is amplified with a unique combination of the barcodes. Miniaturization of this step requires a low-volume dispenser. And this is a screenshot of the Assay Studio, the software that comes along with the iDot. It shows an example for barcoding 96 libraries. On the left, you see the source plate loaded with the 12 forward and 8 reverse primers from the Illumina kit. And on the right, you see our 384 well target plate with the 96 segmented cDNA libraries that we want to index. A nice feature of the iDot Assay Studio really is that it allows you to run a simulation prior you actually start the dispensing run. And as shown here, the time needed to index 96 libraries is 1 minute and 5 seconds. And this can be even faster if we further reduce the volume dispensed. Here we actually used 0.5 microliter of each primer. Given that up to 96 primers can be loaded onto the iDot source plate, one can barcode more than 2,300 libraries at once. And obviously, any cross-contamination between the primers has to be avoided, since it would make proper demultiplexing impossible. This cannot be achieved with other single or multi-channel low-volume dispensing instruments. Another example of how the iDot can efficiently utilize is the normalization of DNA samples, such as the cDNA libraries obtained in this workflow. This requires to dispense different volumes of buffer or water and of DNA for each individual sample. We did this by loading 96 samples onto an iDot pure plate, dispensing those for subsequent quantification. This is commonly done with intercalating dyes using a plate imager. Here we used an assay called picogreen at reduced volumes. The results from the plate reader can be then automatically translated to a CSV file or an Excel table that we can load to the iDot. Within minutes, the iDot can be then set up to dispense and normalize 96 cDNA libraries in parallel, as you can see on the slide. For setting up the RNA-seq workflow I presented today, we used a well-characterized cell line. Most likely you're interested in looking at more complex samples, and this slide highlights that our single-cell dispensing technology is really compatible with a wide range of different samples. Here you can see nozzle images generated by the F-site from various cell lines, including CRISPR-Cas9 edited stem cells, but also for primary cells such as hepatocytes, neurons, and cells collected from blood samples. For smaller cells, such as yeast and bacteria, the B-site instrument can be integrated into such a workflow in a similar manner. Today, I talked a lot about SmartSeq2. In a similar manner, other single-cell assays can be set up on our platform. For example, we have customers that use our systems for variations of SmartSeq, such as the recently published protocol for SmartSeq3. In a similar manner, other plate-based methods can be also performed, such as QuadSeq, CellSeq, or multi-omics methods like GNT-seq. In case you're working with bulk samples, or if you use FACS sorting to isolate your cells, the iDot is a great tool to miniaturize the reactions and the library preparation. I'm now almost at the end of today's presentation, so let me summarize the key features of our instruments that we used for this workflow. Using image-based cell detection, the F-site allows for efficient deposition of cells into conical PCR wells. You can expect a throughput of 2 to 5 minutes per 96 well plate. It comes with a fluorescent laser to sort cells based on fluorescent markers. And the technology is clearly more gentle compared to flow cytometry, and you can start with very small amounts of sample volume to isolate cells from. The iDot is really one of the most versatile low volume liquid handling technologies on the market. It features a low dead volume of less than 1 microliter, and it allows to load and dispense of up to 96 reagents in parallel. It is faster than comparable acoustic dispensing technologies and comes at a fraction of the price. Both instruments are easy to use and can be set up within minutes. By combining the two instruments, you can get an affordable and highly flexible platform for single-cell omics analysis. The instrumentation is compatible with basically any plate-based assay. The workflow is entirely contact-free, which saves a lot of pipette tips. Single-use cartridges and single-use dispensing plates eliminate the risk for cross-contamination between samples and provide an ideal basis and workflow for working with clinical samples. The multiplexing capability of the iDot allows for rapid sample barcoding, as I presented earlier, and both instruments come with an automation API that makes the integration into a fully automated workflow straightforward. We are happy to share protocols and also support you in establishing new assays and methods on our systems. In case if you're interested in learning more about the technology, there's a few things that we offer. We typically are always able to set up individual telecons to discuss your application, your workflows. After such a telecon, we would then proceed with an on-site demo to introduce you to our instruments and also let you work with your samples, with your assay on our instruments. Due to travel restrictions, we now have set up a live demo room in our labs. So if you're interested, now it's really the chance to get an application-specific live demo that you can watch from anywhere. And this can be also done on a very short notice. So please get in touch with us, and our team is ready to prep a demo for you. Now we are at the end of today's presentation. This was the third webinar of our CYTENA webinar series we hosted in April. If you are interested in the other applications, cell line development and the isolation of microbial cells, please visit the CellLink website where the recorded presentations from the last 2 weeks can be downloaded. There's also more webinars and more material on other technologies we offer within the CellLink family. With that, I would like to thank you for joining. I hope you enjoyed the presentation, and I'm now ready to take your questions. Okay, there's quite a few questions that came in. There's one question which probably came up after I showed the video with the single cell dispenser. The question here is whether the dispenser moves or the substrate, and actually in the Excite series, the dispenser is kind of static, and we move the substrate below the dispenser to move from well to well. How could the dispenser differentiate a single cell with debris or artifacts? That's a good question. So what we do is we do cell detection based on bright field and fluorescent imaging. This allows us to really extract a variety of features from these images, including the shape of the cell, the size, the roundness, but also the fluorescent signal. And using these parameters, we can make sure that we only sort and gauge for single cells, and using, for example, the roundness criterion, we know that we can select for viable cells, which can be in addition supported by labeling them with a fluorescent marker. So there's different things you can do to sort for single viable cells. Typically, bright field imaging is sufficient to do so. There's a question on how the IDOT is calibrated. I think it's probably a good idea to have a call to go a bit more in detail here, but this can be actually done by our customers, and there's no special reagents or a third-party tool needed to do so. What I didn't mention today due to time restrictions is that the IDOT comes with a droplet verification tool that really counts the number of droplets that are dispensed during each dispensing step. And this serves not only as a quality control that lets you check whether everything was dispensed as outlined in the protocol, it can be also used to calibrate the instrument on site with new reagents with a very simple procedure. There's another question on the IDOT, whether the liquid might leak out of the hole in the bottom when there's a pressure applied. So the nozzles of the IDOT at the bottom of the wells are quite small. There's different versions. They're typically in the order of 90 to 100 microns, and surface tension guarantees that the liquid is not leaking out of these holes, and only when the instrument really applies a well-controlled pressure pulse, droplets are generated by the instrument. There's a question on the chemistry. I've to use glycerol in my protocol. Can I dispense reagents with higher viscosities with the I-dot? Yes, that's possible. We actually tested glycerol, and we already demonstrated that up to 25% glycerol dispensing can be done accurately. And there's other reagents that are frequently used in these assays. For example, SmartSec3 uses PEG as a molecular crowding agent, which also increases the viscosity. We therefore also confirmed the dispensing performance with PEG. For example, PEG 8000 can be reliable dispensed up to 12%, and therefore the I-dot is compatible with these protocols. How does the sorting of the cells work? Is pressure applied? What's the limitation in cell size? That's also a good question. I didn't go into too much detail today. In the other two webinars, I presented a few more slides on how we actually generate droplets with single cells and how we make sure we only dispense single cells. In brief, we generated droplets with a piezoelectric drop-on-demand dispenser that is somewhat similar to the ones utilized in inkjet printers. So there's no pressure involved, which really results in a very gentle process. And we make sure that we only dispense the droplets with single cells to the target substrate by removing the droplets that we do not want to dispense using a vacuum suction. I think with that we are at the end of our session here. We are happy to address further questions via email or phone calls afterwards. Please get in touch with us if anything comes up, and check out the other webinars. There's more stuff upcoming this and next week. Thank you very much for your attendance, and have a good rest of the day.